Are you a pharmacy aspirant? Are you thinking to get more support beside your classroom? Then stop worrying about all these now, because Pharmacy Guide has come now at your service. If you want to join our team as a teacher and want to share your knowledge with all out there, please email us at pharmacyguide7@gmail.com.
Hello everyone, welcoming all of you to Pharmacy Guide. Myself Sakshar Shah, I'm an assistant professor from the Department of Pharmaceutical Technology, JIS University, welcoming all of you in today's video lecture. So in this days we have talking about the cell culture and different aspects of cell culture, and today the topic I'm going to discuss about, it is very important in case of cell culture, not only cell culture, in a separate way as well. So the topic is flow cytometry. Flow cytometry altogether is a very big or very important topic. So what I tried to do to give an overview of it, thus you can understand the phenomenon pretty well, but I'm not giving you a detailed view of it. Okay, so before moving on to my moving on to in detail, I'm just telling it to you that it is not a detailed study or detailed lecture I'm giving it to you. Thus you will understand an overview of it. So that's why this video is being made. Okay, so flow cytometry is basically a phenomenon where you can analyze a cell, or you can analyze the physical or chemical property of the cell. You can do the qualitative analysis of the cell. You can analyze its physical characteristics, you can analyze its chemical characteristics. So in case of flow cytometry, if you talk about the principle of flow cytometry, then it would be it is a detection or is analysis of the cell qualitatively by its physical and chemical structure or characteristics, where cell suspension has to pass through a laser beam, and due to that laser beam, that cells from those cell suspension used to scatter some of the beam, some of the light, and those light are being collected and detected the cell structure or cell's morphology. So this is the basic principle in case of cell cytometry, or this is the way the cell morphology can be studied. So in case of, if we talk about components, so there is dichroic mirror, there is filter, detector, laser, fluidic system. Dichroic mirror is basically used to collect the scattered light and used to send according to their color. Okay, send to the detectors according to their color. In case of filter used to filter out those specific wavelength light. Detector used to receive the light which is coming from the filter. That's why from filter to detector this arrow is being given. Laser as in laser beam, the source of light, and there is fluidic system. So in the name itself, it is written flow cytometry. Cyto means cell and flow. So cells are in flow. So if it is flow, that means that some kind of fluidic system or some kind of fluid will also be associated with it. So this fluid system is basically two types. One is the system. I'm not saying the fluid. The fluidic system is two types: central channel and the outer seat. Okay, I'm just interrupting that. I'm sure you are somehow thinking that, oh my God, what he's saying or something like that, because I'm just talking about the components. Okay, so this is the simple diagram I tried to draw, though it is not so good. But at the end of the video, I will show some of the picture, very nice pictures, and I'll describe. So I'll request all of you to keep watching till the end, so you will understand this whole phenomenon better. Okay. So fluidic system is having two parts. One is central channel, where the sample are there. There is outer seed. Okay, that where the seed fluid are present. Seed fluid as in saline or PBS. 0.9% saline can be present, or PBS can be present as a seed fluid. So so here the sample as in cell suspension. Here the pressure in the central chamber where the sample is present, the pressure have been denoted by PC, and the outer seed where seed fluid is present, the pressure there is being denoted by PS. So in PC is generally used to be greater than the pressure in the central channel in the sample used to be more than PS, that the pressure in the outer seed. So it is which denotes which confirms the high flow rate, and high flow rate is required in case of immunophototyping. Immunophototyping is one of the application of flow cytometry. But in case of DNA analysis, the flow rate used to, the required flow rate used to be less. So that time, PS used to be greater than PC. That means PC, the pressure in the central channel or in the sample used to be less than the seed fluid. In case of DNA analysis. DNA analysis you can also do by the use of flow cytometry. So from here you know that there is at least two application of flow cytometry is DNA analysis and immunophototyping. So let's move on to this diagram. So sorry if you're not understanding, because this is the best I could have done. So as I have spoke about fluid system, so this whole structure is basically the internal structure of the flow cytometer machine. Okay, so it is, you cannot see it from outside. From outside it looks completely different. It's an inner view of that equipment. So the central channel where the sample is there, this one is the central channel, and this one is the CC as in central channel, and this one is the outer seed, OS. So in the OS, the saline, 0.9% saline or PBS are flowing, and here the cell suspension is there. If the concentration of the cell suspension is low, so that is quite good. The concentration of the cell suspension should be lower. It should not be very viscous. And here one important point is coming, that is called hydrodynamic focusing. Hydrodynamic focusing means, what is the reason that this outer seed fluid system is there? Because this is a cell suspension. So here the flow cytometry, in the principle of flow cytometry, says that the single cell suspension should pass through a narrow channel, which makes sure that from that narrow channel only one cell is passing. Okay, so you can see from this narrow channel only one cell is passing. From the narrow channel, it is so tiny, it is so small that only one cell will pass through. And this force, the flow from the outer seed, will ensure that it should go. It is an influencing factor to maintain a streamline, equal flow of the cell suspension. So that's why this region is being called hydrodynamic region, hydrodynamic focusing region, and this phenomenon used to be called as hydrodynamic focusing. So here the cell is coming, and from here the laser beam is there. From here the laser beam is there. Laser beam is the beam is going in this direction. So the point where one single cell is touching the laser beam used to be called as interrogation point. Interrogation point. So from interrogation point, it is going to the PMT. These are called, this arrow portion is being called PMT. Photo multiplier tube. Sorry, PMT I have wrote. Sorry, it is called PMT. Photo multiplier tube. This 1, 2, 3. This 3 is called photo multiplier tube. And this 2 is called dichoric mirror. The dichoric mirror I have spoken. The dichoric mirror, what it does, okay, before moving on to that, let me tell you. So when the beam is touching this single cell in the interrogation point, the cell used to scatter the light. So there is two types of scatter. One is forward scatter, another one is side scatter. Forward scatter is termed as FSC, the side scatter is termed as SSC. Okay, so forward scatter determines the cell size. Okay, the side scatter determines the cell's complexity or the structure or the cytosolic structure, the cell's internal structure, cytosolic structure or complexity determined by the side scatter. So inside the cell there could be different components and that could be of different color. So to determine those colors, this dichroic mirror is there. Maybe I'm just giving an example, this dichroic mirror is collecting the red color components picture. This one is green, this one is a blue, or some, something different thing, and which is being transmitted to PMT, photomultiplier tube. The function of photomultiplier tube is to collect the photons which is coming from this dichroic mirror and transform or convert it to electronic signals. And those electronic signals in going from this forward scattered and from this side scattered, it is going to ADC. This one I wrote ADC. ADC full form is analog to digital converter. So it is collecting all those electronic signals and it is converting it to the digital content and it is sending to the computer. And from the computer we are seeing some of the picture. So this is the basic phenomenon inside of flow cytometer happening. So we used to get two types of picture. One is from FSC, that is forward scattered light. Another one is from SSC, that is side scattered light. In a side scattered light, as you can see here, if we can merge those two pictures together, forward scattered and side scattered light, this kind of histogram we will find out. So here in the the x-axis we have plotted FSC, in the y-axis there is SSC, side scattered light. So this compound, this particular compound is having a high value of FSC and high value of SSC as well. FSC denotes the cell size. So this particular compound is having a very big cell size. And this SSC denotes the complexity. So the internal structure should be something which is causing obstruction to reflect the cell, right? Because side scattered light denotes the complexity. The much obstruction it will face, it will show more scattering. So one more thing I am telling you, if the scattering angle is very high, if the scattering angle, the side scattering angle is very high, then it is having, the component is having high complexity inside it. The lower scattering, like the lower angle light used to be called as FSC. The higher angle scattered light used to be called as SSC, because it used to get in this way. It used to scattered in a wide range. So if the scattered light is having a high angle near to 90 degrees, then you can confirm, you can assure that that particular compound, that particular cell is having a very complex structure inside it. So this could be a granulocyte, because there are so many granules inside the cell which is causing the obstruction, and that's the reason it is having a high SSC value. This particular component is having a high FSC value, but it is lower in SSC. This could be compound such as monocyte. This one could be lymphocyte, and there is a very small value of FSC and small value of SSC. So there is tiny structure, that could be RBC, or that could be some dead cells or cell debris. All those fragments kind of things could be this one, which is having low FSC and low SSC value. So this was all about a very basic discussion, very basic discussion about flow cytometry. Though the video hasn't ended yet, after I take the leave, there will be a small demonstration. You would see there, you can see some of the nice pictures, though it is not. So I will request all of you to stay up to that and see the part, so you will understand the flow cytometer much better, I think. So I'm taking the leave from now on. I hope you are understanding my videos. I'll request all of you to subscribe my channel. If you have not done yet, I'll request all of you as well to share this channel with your friends, colleagues. I'm sure they will also be liking it or also be benefited by this one. Put in the comment section what you are thinking about my video, what you want to see, what kind of topic you would like to have a lecture video. I will really try to come up with that kind of topic, but your comments, whether it is good, whether it is bad, is really important for me to improvise in my video to improve myself. Thank you so much. Thanks for watching. Thanks for subscribing my channel, Pharmacy Guide. Do not go right now. The last part of the video is still there. Bye-bye.
So now see some of the nice picture of flow cytometry. I have already discussed about the flow cytometry and its applications and the components related to it. So here, okay, let me just point here out. So here you can see some of the this is the seed fluid, that is which is basically maintaining the hydrodynamic focusing. See, hydrodynamic focusing cell pass through in single cell line, because this hydrodynamic focusing phenomenon makes sure that the cell should pass through in a single line and one cell should pass at a time through that narrow channel. Thus, one cell should get in touch with that beam. Here you can see the laser light source. There is a laser beam which is coming. This is a laser beam, again green color, and it is touching the one cell, and this point is being called as interrogation point. And the forward scatter, as you can see, the green line is the forward scatter, and SSC is the side scattered, is happening with the fluorescence emitted from the stained cells detected. So forward scatter, so fluorescence emitted, because some of the, as I said, that some antibodies, there is red fluorescence antibody which is being attached. Those are the cell marker or cell internal cell marker, which used to be attached initially during the preparation of cell suspension, used to get attached to the has been added, used to be added additionally to attach to the cell, that the fluorescence emitted can emit after the collision with this light source beam that is leaving, and the fluorescence which should be emitted, and that should be detected by the different dichroic mirror, and the internal cell structure or cytosolic structure can be determined. So here the total the whole structure which I tried to draw in the board, but I could not in a very proper way. As you can see, the cell sample, the laser beam, the single cell is coming, and from this part there is that a seed fluid, but that is not being shown here. That the dichroic mirror, what it is doing, dichroic mirror is this channelizing the light, and this is that uh, filter which is filtering out the specific color light. So this one, red, green, blue, all these are PMT. There is obscuration bar basically what it does, it's basically allows maximum amount of beam towards one side, or if it is a very less amount of beam, then it makes it a very wide angle. If it is a very wide angle beam, it makes it a exact angle beam. So here it is making some of the changes, alteration, and it is sending that beam which has been scattered forwardly from this particular cell to the PMT. PMT is photomultiplier tube. The function of it is basically transferring the photons to the electronic signals, and all are going to ADC, that is your analog to digital converter, and that digital content is being shown in this computer. So now this is the external view of flow cytometer. So this is the way, this is how it looks. It's fully a sophisticated apparatus. It looks like a sophisticated apparatus, and it is a sophisticated apparatus. And I'm sure you have noticed that this part, okay, this portion where you can see a glass tube has been inserted. That is the part from the outside of the apparatus a cell suspension used to be given, and there is a pipette like thing which used to suck it inside, and all the procedure which I showed in the previous part which used to happen inside this apparatus. So this is all about the flow cytometry and the things related to it. I'm sure you have understood because I tried to give a basic idea about flow cytometry, though it's a vast topic. I did not explain the detailed view of it. Okay, so this is all about flow cytometry, and in the next video I'll try to come up with a different application of the flow cytometry. I request all of you to subscribe my channel if you have not done. Pharmacy Guide is my channel. I'll request all of you to share it with your friends. And if you're not subscribing, you will not be notified when I'm on YouTube or when I'm uploading a video on YouTube. So, and put in the comment section whether you are liking my video or not, or what are the topics you would like to see. You would like me to come up with those topics or make a video lecture on this. Let me know. I'll try to come up with all those things. Thank you so much. Thanks for your support. Keep watching Pharmacy Guide. Bye bye.